Yes, I hear you. Can you hear me?
Yes, I can. Thank you.
Wonderful.
Okay, so we're going to start the session with a talk on the difference between HPV DNA and HPV RNA assays and clinical evidence by Professor Thomas Ifner, who's a professor of virology at University Hospital of Tübingen, Germany. So, Professor Ifner, over to you. If you can share your screen and slides that you would like to show.
Okay, can you see my screen? Yes, okay, wonderful. So, first I would like to thank the organizers for inviting me to this cytopathology tutorial in Lahore from the International Academy of Cytopathology in Pakistan. As the chairman said, I'm from Germany. I'm a virologist, but I also take care of the National Institute of Virology at the ICCBS at the University of Karachi in Pakistan. So I'm somehow linked to your country. So thanks for inviting me. And if you have any questions, please feel free to ask me the questions after the talk. Okay, so worldwide, the cervical cancer incidence, so cancer of the cervix uteri is quite large, with almost 600,000 new cases a year. And when it comes to absolute numbers, you can see the highest counts are in South Central Asia with regard to incidence and mortality. When we look now for Pakistan, the numbers published by the WHO, if they are correct, are quite good, because Pakistan is, when it comes to the incidence of cervical cancer, in the lower part of this figure, where India is much higher, one of your neighbors. But when it comes to mortality, it looks like the trend is actually going up, and it's not like, for example, for South Korea, where they have a higher incidence but a lower mortality. And this is a kind of an indication that in Pakistan, probably most of the cancers are detected at a very advanced stage, and where there are no really good therapeutic options anymore. Okay, now HPV has been shown without any doubt that the persistent infection with the so-called high-risk HPV type is a necessary risk factor for cervical cancer. So if you remove this necessary risk factor, you will not have cervical cancer. And the few cases of HPV negative cervical cancers are mostly misclassified cancers by pathology and not primary cervical cancers. And this is a justification for the development of tests to detect the presence of HPV, and for also the vaccination that is nowadays available, where you can have a prophylactic vaccine that prevent actually to be infected with those high-risk types. Now, papillomaviruses are small DNA tumor viruses, and they have no lipid envelope. There are more than 200 types that affect humans. They are strictly species-specific. Very importantly, they only replicate at the local site where they enter the body, and they don't make any systemic infection. And the inside of the capsid, there is a double-stranded DNA of about 8,000 base pairs. And the only thing you need to know is there are two genes that encode the viral oncoproteins E6 and E7, and they are regulated by another viral gene called E2. Already so not all of these 210 types are share the same risk. And already in 1995, the WHO has classified two types, namely HPV 16 and HPV 18, as carcinogenic for humans, because 16 and 18, those two types cause about 70% of all cervical cancers worldwide. Then 2005, there were another types, 10 types added, that are shown here. In 2010, HPV 68 was also added as a class 2A carcinogenic type. So in total, we have 13 types that are called high-risk human papillomavirus types, in contrast to the low-risk types like HPV 6 and HPV 11 that cause genital warts. Now, fortunately, cervical cancer is a cancer that takes some time to develop, and it does not develop de novo. but it goes through precursor stages that are called cervical intraepithelial neoplasias, and they are graded from CIN1 to CIN3, and that can progress into invasive carcinoma. There is a big difference between CIN1 and the other two groups, CIN2 and 3, because CIN1 can actually caused by low-risk types and by high-risk types. And in CIN1, it's very important, the virus cycle can still go through and make new virus particles, so we have a productive infection. However, in CIN2 and CIN3, there is an abortive infection going on. No longer virus particles can be produced, so the viruses actually are catched in this epithelium. Good news is, again, it takes up to 15 years that these cancers can develop from the infection of the normal healthy cervix. And there is still a possibility of spontaneous regression also for CIN3, although this is very low. But for example, CIN2 regresses within a few years in about 50% spontaneously. And this was the success story, actually, of this cervical cytopathology, the pap smear to detect precursor lesions that has been implemented in most countries around in the 1970s. And this is done by, usually you take a cytobrush and you rotate it for 360 degrees, and thereby you take cells from the endocervix and from the ectocervix. You smear the cells on a glass slide. They are stained according to Papa Nicolau, and then they are examined with the help of microscope, and eventually you can detect abnormal cells like here. However, it has been shown in the last decades, really, that a single smear has only a sensitivity of about 50% to detect CIN3. And this was the reason why a while ago also we started trials to compare the performance of pap smear with HPV testing in large cohorts. Here you can see this is a study where 6 countries participated with 25,000 women, where women were tested either by pap smear and by HPV testing, and followed up for about 7 years. And then we looked for the performance of the 2 different tests. And we could already at that time, 2008, show that women that were negative at baseline and had a normal pap smear have a much higher chance to develop afterwards CIN3. This is a cumulative incidence of CIN3 per 10,000 women on this axis here, as if the women were either negative by the HPV test or had a normal smear and were negative by the HPV test, where the curve is much more flat. So the risk of those women is much lower to develop later on CIN3, whereas a negative pap smear was not really predictive. And this was the reason why randomized control trials started in different countries in Europe, in Sweden, Netherlands, Italy, and UK. And now they used as endpoint cervical cancer. A total of 176,000 women was included. Again, you see the cumulative incidence of cervical cancer here on the y-axis, and you see women that were at baseline had a normal smear or were HPV negative for high-risk HPV types, and after 3 years the curves split up, as you can see, and women with a normal pap smear have a much higher risk of cervical cancer than women that had an initial HPV negative test. And this is now also followed by real-world evidence data, not trials. So I show you just one slide here from the entire state of New Mexico, where they followed up the performance of their cervical cancer screening program that is cytology-based between 2014 and 2017. And actually, it was not only cytology-based, they actually 67% of the tests were performed as co-tests, where they did HPV testing in combination with cytology, and they used an Different HPV tests, as you can see here, 60% of the tests reported were either the Roche Cobas test, the hybrid capture test by QIAGEN, or the RNA-based test by Optima Biologic. Endpoints were CIN2 and 3, and you can see when you add HPV testing to a cytology-based cervical cancer screening program, you get 21.9% additional CIN3 cases that were HPV positive but cytology negative, with a modest increase of the 2-year biopsy rate from 3.2 to 4.8%. And this is a very safe approach. As you can see, only 3.2% of CIN3 cases were missed by this approach by co-testing. And it's also very good predictive for women that were negative by both tests, because only 17 women in the entire state of New Mexico developed CIN3 plus within 2 years. And this is approximately 1 in 10,000. So showing human papillomavirus testing for cervical cancer screening increases the diagnosis of precancer and reduces the incidence of cervical cancer more than if you would have done cytology alone. However, it is very important to keep in mind that in the younger age group, the prevalence of high-risk HPV types is very high. And these are data from Germany showing that when you age stratify the HPV prevalence in young women, you see a peak in women at the age of 20 to 22, where 24% actually were high-risk HPV positive, and this goes down starting from the age of 30 down to 5 to 4%. So meaning, do not make an HPV test under the age of 30, because you would send all these women that probably have transient infections and not persistent infections and will not develop cervical cancer to unnecessary follow-ups. Now, how can you detect HPV? So there is a possibility to detect the DNA that is inside of the viral particle and is also present in the cells that are infected with a DNA-based test. And from that DNA, the virus needs to transcribe messenger RNA to replicate the DNA and to actually transform the infected cell. So a test that is based on messenger RNA will detect the active virus infection. The WHO has a few years ago published their global strategy towards the elimination of cervical cancer, with a vision to have a world without cervical cancer, defined as all countries should reach less than 4 cases per 100,000 women years. And to make this able, they set control targets to be reached in the year 2030, very ambitious goals actually, but meaning that 90% of girls should be fully vaccinated with HPV vaccine by the age of 15. 70% of all women in those countries should be screened with a high precision test twice in their lifetime, at the age of 35 and at the age of 45. And it makes no sense to do this if you do not follow up these women then afterwards, when they are positive, to give them the appropriate treatment. So they defined 90% of women that have an identified cervical disease should receive treatment and care. However, you cannot just use one of the tests that are able from the shell, or you can order in the internet, because a recent review actually showed that at the meantime 254 HPV tests are present on the global market, and this was 2020. However, when you look at these HPV tests, you see that almost 60% of them have not a single publication. So there's nothing known about those tests. In addition, 82% of those tests have no published analytical or clinical evaluation in peer-reviewed journals. And more than 90% of those tests actually do not fulfill the standards that are agreed on in the HPV community that those tests should fulfill. So I will restrict my talk to those most commonly used HPV tests that fulfill the requirements that have been set in the HPV community that a test should fulfill to be able to be used for screening for cervical cancer. And these are DNA tests and the RNA test, Optima. They have different targets, either the whole viral genome, the E6, E7 encoding region, so the ones, the genes that encode the viral oncogenes, or a major structure, the gene that encodes the major structural protein, L1, which is also used for the vaccine. Some of them can also identify specific genotypes. Mostly they are consensus tests that can give you an answer if there's a high-risk infection, yes or no. Different techniques are used. A lot of them use real-time PCR. Some of them have an internal control gene for human genes to see if the reaction is working. And most of them actually are approved by the FDA. And this is very important. You should have an approval by an authority that makes a strict, that has strict requirements that such a test can be used for cervical screening. And when you now compare the kappa values of those tests from different studies to HyperCapture, which is a reference test actually on the market, or was on the market, it's more or less dropping out. Then you can see they have all a quite good comparison. And when you look in another meta-analysis where they look for the performance of these tests relative to the HyperCapture with regard to sensitivity for CIN3+ or specificity of less than CIN2, you can see those tests perform very similar. And now I want to show you some data about how good DNA and HPV messenger RNA tests perform accordingly. The WHO recommends to use HPV DNA, nucleic acid amplification tests, but also HPV messenger RNA detection may be used, they state. Again, what is the difference between a DNA and an RNA test? So the DNA test can actually also detect viral particles outside of a cell if you have a protocol that lyses the viral particles. It can also pick up cells that have been infected but in the meantime are dead, but there is still DNA present from the virus, but also they detect living infected cells. In contrast, the RNA test is only able to detect an active infection in a living infected cell. However, those cells are not single cells, where those cells are always within a stratified epithelium, within a connective tissue, as you can see here in a schematic drawing of the cervical epithelium here. And I have also given you the respective histopathological pictures for CIN1 and 2 and 3 above. So because it's a very simplified drawing here, in this drawing you can see the cells. Yellow stars indicate the presence of viral DNA, and the size correlates with the amount of DNA. Purple cells actually show you abnormal cells that are infected by HPV, and they express, all of them express viral RNA, and they also have viral DNA in different amounts actually. And what you can see here, when you go from CIN1 to CIN3, you can see in CIN1, look at the histopathological picture above, you can see it looks almost like normal epithelium, only in the suprabasal. There are only a few suprabasal cell layers that still can proliferate, whereas in normal epithelium, proliferation only takes place in the basal layer. However, the epithelium shows normal differentiation going on, and this is very important because the virus replication cycle is coupled to the differentiation of the epithelium, and only in highly differentiated cells the virus can amplify its viral DNA in infected cells up to a few thousand copies per cell, and then those copies can be packaged into new viral particles, and the particles are released from the surface of the epithelium. So in a CIN1, you have, when you take a biopsy from this, you have a lot of viral DNA because of this high amplification. In the basal layer, usually have only 50 to 100 copies because there is no packaging and no amplification of the viral DNA, but only a replication where you need viral genes expressed. So the messenger RNA of the virus is always present in these cells. And so when you look at the cells, probably the chance to be picked up by a DNA test is much higher than by a messenger RNA test, because there's relatively few messenger RNA in comparison to a lot of DNA in a CIN1. In a CIN2 and 3, the amount of abnormal cells can make up to the full epithelium, as you can see in CIN3. So you have a lot of abnormal cells with messenger RNA. However, the epithelium has lost the abilities to support the productive infection. So there is no viral DNA amplified, no virus particles are produced. So in these two stages, CN2 and 3, we have a loss of viral DNA and an increase of viral messenger RNA. And in addition, the viral genome very often integrates during the progression of the lesion into the host chromosome, and thereby you have again a loss of viral DNA because usually have the integration of only a few copies. So during the process of progression from CIN1 to CIN3 and cancer, you have a decrease in HPV DNA and an increase in HPV6 messenger RNA. So the messenger RNA test is much more likely to pick up higher grade lesions than low grade lesions, and this is very important. And now I'm going to show you the results quickly of the German Aptima study, where we co-tested women with HPV and cytology, and we actually also compared the performance of the RNA test Aptima with the performance of the hybrid capture DNA test. And this was all done from liquid cytology tube. So one smear was taken from the women, and from one smear, both DNA and RNA test was performed, cytology was performed, and in case of a positive HPV test result, we also genotyped the HPV type that was present. Though this is a study protocol, 10,000 women aged 30 to 60 were invited in a multi-center study in Tübingen at 3 places. Exclusion criteria are shown here, and we invited positive tested women either by cytology or by HPV, messenger RNA or DNA. So in any case of a positive test result, they were invited within 8 weeks to colposcopy. We also invited 5% of women that were negatively tested in any one of those tests as a control group. And women, we also had a follow-up that I will show you. So positive tested women who had no therapy after colposcopy and histology were followed up yearly for up to 5 to 6 years with the same procedure. And after 5 years, we also invited women that had a triple negative test result to be retested with the very same procedure. And I show you now the results of the cross-sectional study. So what you see here is the sensitivity of the test from 0 to 100%, and on the x-axis 1 minus specificity. So the ideal test with 100% sensitivity and specificity would be here. And as you can see here, there is no difference in the sensitivity between the hybrid capture DNA test and the Aptima RNA test. Sensitivity was very similar. Also, the confidence interval for the RNA test was higher. However, specificity of the RNA test Aptima was a little bit higher than the hybrid capture. And this minor increase in specificity actually leads to a 20% reduction in the referral rate. So by far less women had to be sent to colposcopy based on an Aptima RNA-based test result than women based on a DNA-based test result. And as I said, we had a follow-up of those women, those 10,000 women. Again, the triple negative cohort was invited after 5 years, and we performed again co-testing. And all women that stayed positive and were not treated were invited on a yearly basis. And whenever they had a positive finding, they had to go to colposcopy. They dropped out when they had a positive biopsy for a CIN3 and had a conization. If not, they were retested after 1 year. And the crude results of this follow-up is shown here. Here you see the triple negative women, which is a majority. We invited 4,000 women for follow-up. 3,295 actually followed this invitation, and we had only 4 CIN3 lesions in these women that were triple negative at baseline and 5 CIN2. Now those women that were still positive and had a yearly follow-up. We had 606 that were eligible, 79% attended the invitation and followed the invitation, and those women that were positive by cytology and HPV at baseline, that were 60. We had 6 CIN3 in that group. Interestingly, out of the women that had at baseline only an abnormal cytology but a negative HPV test result, we had not a single CIN2 or higher in this group. And most of the lesions were in the largest group of women that were cytology normal but HPV positive at baseline, and we had 18 CIN3+. And if we now put all these results together and calculate the cumulative risk incidence of CIN3+ after a negative baseline result per 1,000 women screened, you can see the green curve here is cytology, showing a much higher cumulative incidence rate of CIN3+ after during the observation period from 0 to 6 years. And the blue curve is Aptima messenger RNA test. The red curve is the hybrid capture DNA test. There is no significant difference between those two, and you can see it's much lower again than for cytology. In the meantime, there have been also 10-year follow-up data published where they also compared DNA testing versus RNA testing. This was the focal trial in Canada, a long-term study. They had a control arm with 9,457 women with liquid-based cytology at 24 months, and then a co-test with HPV and liquid-based cytology at 48. The intervention arm, they used HPV at baseline and co-testing at 48 months, and in the safety arm, they used HPV at baseline and liquid-based cytology at 24 months. Now, they did the HPV test comparison in the intervention arm, where they compared the DNA-based test hybrid capture again, but also the Roche Cobas 4800, and again with the Aptima messenger RNA test. And to summarize already at this point, they found significantly fewer RNA positive women at baseline than women for Cobas DNA test, and the risk for CIN2 and 3+ was comparable for DNA and RNA based tests. And this is shown in this Kaplan-Meier curve. Because there were no tests performed in the first 48 months after baseline for those that were HPV negative, the curve starts to go up for the single cases. Here's the comparison of the two DNA tests at 4 years, and here are the absolute numbers. And you can see the confidence intervals completely overlap, and there was no difference between the performance of the DNA test Cobas and hybrid capture. Also, the hybrid capture has no internal control, whereas Cobas has. And here they added also the RNA test Aptima, and again, when you look at the numbers, you see no differences, and all the confidence intervals completely overlap. So there is no difference, and all three tests are completely safe, even for a follow-up period of up to 10 years. Now, very recently, there were also the data published from the English screening pilot. In the UK, what they do, before they make a nationwide screening program, they set up sentinel sites at 7 places where they started a screening pilot. So women aged 24 to 49 were called in every 3 years and had a co-test out of liquid-based cytology and an HPV test, and women 50 to 64 had the same at every 5 years. And you see the different sites use different HPV tests. So they use the Aptima RNA test, they use the real-time Abbott test, and they use the Cobas, the Roche Cobas test. And here are the results from the first screening round. And when you look at the number of CIN3 lesions detected per 1,000 women screened by the RNA test, by the Abbott DNA, and by the Roche DNA test, you see there is no difference in the detection rate of CIN3 between all three tests, also shown by the odds ratio here. And in the second screening round, first note, by far less women are now positive and have a CIN3, and this is always seen when you start including HPV testing in a cervical cancer screening program. In the second round, all the women were actually fished out in the first round that had abnormal lesions, and in the second round you only see the incident cases. But when you look again for the number of CIN3 plus lesions detected per 1,000 women screened, and you compare the three tests, you can see again no difference. Very importantly, in this study, for the first time, they also published the number of interval cancers that are diagnosed between the first and second screening round. And this is now also split for the three different tests. However, they put the COBAS and the real-time DNA test together. And when you look now over the observation period for up to 70 months, you can see the curve. The blue line is the COBAS and the real-time DNA test. And you see there are interval cancers showing up. And the orange line is the Aptima messenger RNA test. As you can see here, and here are the numbers of cervical cancers diagnosed per 100,000 women years. And you can see there was one case for Aptima, 4 per real-time, 8 for COBAS, combined 12 of those two. And when you now calculate the hazard risk, for sure there is no higher number of interval cancers when you use the Aptima messenger RNA test. Here it looks even as it is reduced. And again, this comparison of the performance of messenger RNA tests in comparison to hybrid capture DNA test was also published in a systematic review and meta-analysis by Mark Arbin. And as you can see here, for the Aptima test, relative sensitivity is the same as for hybrid capture, whereas the relative specificity is higher, which is significant. And again, this because of that, you have a lower number of women that need to be referred to follow-up procedures. The other HPV tests performed by far, the other HPV messenger RNA tests performed by far less good than the Aptima test. And this is also shown here in the summary table by Mark Arvin in the same review. For Aptima, they show very same sensitivity for CIN3 plus than DNA tests, however, higher specificity. And Mark Arbin concluded that Aptima could be accepted for primary cervical cancer screening on clinician-collected cervical samples, even at intervals of around 5 years. Now, what about when you have an HPV positive woman, however, you have a normal cytology? And this question was addressed by a group in Turkey where they looked at the performance of the Aptima messenger RNA assay and the COBAS DNA platform in women with normal cytology and positive high-risk HPV result. 2,900 women with normal cytology and high-risk positivity were referred for colposcopy. In 134, they found target lesions. And of those 134 patients, they were tested with Aptima and with COBAS, and the positive predictive value of the Aptima was with 36% higher than for the COBAS with 23.4%. So in conclusion, the Aptima assay had a much lower false positivity rate than the COBAS platform, and this is really important because it helps to avoid unnecessary colposcopic examinations which affect cost efficiency of a screening program. Now, what are the barriers now to implement HPV testing in cervical cancer screening? And this is these are major factors in developing countries because there is very often a low awareness of HPV, a poor understanding of the link between HPV and the development of cancer. There are some cultural issues. There's a poor screening uptake, and the follow-up is not good usually. Also, when it comes to provider and facilities, often you find insufficient training, education, insufficient resources and facilities. And if there are no advanced therapeutic options, then it makes no sense to screen at all. And also, if people have difficulties to access healthcare, if there's no insurance or no surveillance and tracking system, then these are real high barriers for implementation. So to sum up, co-testing is a little bit more safe approach than if you do HPV testing alone. This was also shown by a study from Kaiser Permanente, North California, with 1 million women that they screen for a long period as part of their normal screening system, because Kaiser Permanente is also an insurance company, but has also a large number of hospitals. Here you see again the cumulative risk for CIN3+, and the time since a negative test. Cytology again does not have a good as good as a predictive value than HPV, but when they did co-testing, and because of this large number of women, they had a little bit better performance actually, and a little bit more safety. And this is again shown in again from Kaiser Permanente, North California, where they, in this slide here, when they looked at all cervical cancers and up to 6 years prior to diagnosis, and the red area shows women that were HPV positive and cytology positive. The orange women were women that were only HPV positive but negative by cytology. The green area is women that are HPV negative but positive by cytology only, and the blue area is HPV negative, cytology negative women. And when it comes to the detection of the cervical cancer, the closer you get, the higher the areas get here on the scale. And you can see when you do code testing, when you do HPV testing only, you get 90% of all cervical cancers up to half a year before. And when you do code testing, you add another 6% here. So there is still a little bit remaining here, but you never have a 100% efficient cervical cancer screening program. And when you look again at this study here, the effect of several negative rounds of co-testing. So first round, second round, third round here, when you do co-testing here, and this is a cumulative detection of risk for CIN3+, you can see after the second round, you have almost no more risk of detection. And this is better than, a little bit better than when you do only HPV or when you do only cytology. And after the second co-test, there were no more interval cancers detected in that large group. In Germany, we have decided 2020, there was a directive issued on the following screening scheme. And we make a distinction of women that are below 35 or up to 35. So women aged 20 to 34 have an annual cytology screening and no HPV testing because of this high prevalence of HPV, high-risk HPV types in that group I showed you in the beginning. We have a different nomenclature than Bethesda, but I quickly summarize that for you. So if women are normal, they return and they come back after a year. If women have a cytology diagnosis of ASCUS or CIN1, they will be retested with cytology after 6 to 12 months when they are below 30 years of age, and if they are above 30 years of age, this the test is not cytology but is an HPV test. And then if in that case, if the cytology abnormalities persist for 24 months, they go to colposcopy. Or if the HPV test is positive, they go to colposcopy within 3 months. And clearly, those women that have already a diagnosis of CIN3 go within 3 months. And if they have a higher diagnosis cancer, they go immediately to colposcopy. This changes now if women are aged 35 or older. Then co-testing is performed every 3 years, because every 3 years is important. You have to give the virus infection time to resolve spontaneously, and the transient infection may last up to 15 to 18 months, depending on the HPV type. So if women are negative, they return to screening and they are screened again after 3 years. If they are positive in the HPV test and have a normal cytology, or if they are negative in the HPV test and have a CIN1, they have a retesting after 1 year with co-test again. And if they are HPV positive, or if they have an abnormality, they go to colposcopy. And clearly, women that are HPV positive and have higher grade lesions in cytology like CN1 or CN2, they go within 3 months, and if it's higher, they go immediately. So to sum up, when we compare now the performance of DNA versus RNA assays, when it comes to sensitivity, both tests have high sensitivity. Specificity of DNA tests is good, but of messenger RNA tests, it's better. That's why the referral rate with messenger RNA tests is lower than with DNA tests, and the negative predictive value of 10 years is high. So to sum up, only a small number of HPV tests, you have to keep that in mind, are considered as clinically validated for screening. The lower specificity of the HPV test alone dictates the use of triage tests that can select women for colposcopy, and usually this is reflex cytology, because this is currently the only well-validated triage test, and the co-testing is the safest approach, but also the most expensive one. Detection of viral RNA is nothing than a proxy for detection of an active viral infection and for the presence of the HPV DNA in infected cells, and cytology can actually add about 6% additional cases in women with cancer that are HPV negative, as I have shown to you before by the Kaiser Permanente study from North Carolina. Regardless of the study design, similar sensitivities of the RNA and of the DNA test were found for detection of CN3+. This was remarkably stable, and it was also always accompanied by a higher specificity of the Aptima test. Longitudinal studies with RNA and DNA tests show the same long-term risk and longitudinal sensitivity for up to 10 years. And even from real-world screening data are now available, showing sufficient evidence that Aptima tests can be used in cervical cancer screening, even with 5-year intervals. And because of the lower referral rates due to the higher specificity, the test is more cost efficient, and you have for sure, and it's very safe because you have no more interval cancers than if you would use DNA-based tests. And with this, I would like to thank my collaborators on this slide and all the women that participated in the studies. And I would also like to thank you for your attention. Thank you very much.